Hello, everybody. I'm going to tell you about how we've re-envisioned how protein translation works in order to develop a platform to boost yields of recombinant proteins in a platform that we call Codomax. The protein production industry has a large market value of approximately $300 billion, and this is rapidly growing in the coming years. And within the protein production industry, there are many different applications for proteins, whether this is as therapeutics and antibodies, food proteins, vaccine components, or reagents that are used in the biotech industry. However, one key issue is how can we meet this rising demand for protein production, and how can we do so for the lowest possible cost? In order to understand how to approach this problem, though, we first need to look at the fundamentals of how proteins are made in the cell. And the instructions for making a protein are encoded in DNA, and the DNA is transcribed or copied into messenger RNA, where these instructions are fine-tuned and read out into a protein. The instructions in the DNA are called codons. These are blocks of 3 nucleotides that are read one codon at a time to code for the building blocks or amino acids that make up a protein. During translation, which is the process of making the protein from the messenger RNA, the key players are tRNAs. These tRNAs carry one building block at a time for the protein, and they're specific for each particular codon. So each codon is encoded by a particular tRNA that acts as a claw to decode that particular codon. However, we have a bit of a conundrum because there's 64 codons and there's only 20 amino acids. And we've found that this is not an evolutionary redundancy because there's spare codons and spare tRNAs, and there's a secret code to maximizing protein production by choosing which codons and which tRNAs that decode them are optimal. And there are many methods out there currently for codon optimization to improve protein production. These involve looking at the tRNA abundances in a cell that are based on the particular host organism. Metrics also look at minimizing secondary structures in the DNA and RNA, as well as GC content. However, all of these metrics are hypothetical in the sense that they're predictive of the theoretical biology, but not of the actual biology. It's as if you have building blocks with no instructions, and you want to build a model of New York City, you don't have the instructions to do that. We've found that translation's also a bit more nuanced in the sense that you start with a tRNA pool in the cell, but when there's a stress in the cell, whether that's oxidation, alkylation, or starvation, this will reprogram that pool of tRNAs, changing which codons are optimal. So to address this, we've developed a platform where we can look inside the cell and measure the actual codons that are most optimal during the stress of protein production. And we can do this through a combination of omics level measurements, codon analytics, and synthetic biology to measure those optimal codons and boost protein production. This will work where we would have a customer that would have cells expressing protein and it costs them so much to make this protein. We can apply our platform and return to them the cells expressing more protein for thereby a lower cost. As a proof of concept, we turn to one of the major organisms used in industry for protein production, which is Pichia pastoris yeast. And we, as a proof of concept, used Pichia expressing green fluorescent protein, or GFP. And the specific GFP we used has already been codon optimized using the current methods out there today. But we put this through our platform to re-codon optimize the GFP. And we found when we put in the best codons, we get a 2.4-fold increase in yield over the current GFP that's out there. And in turn, if we were to instead put in the worst codons, we get a 10-fold decrease in GFP expression, meaning with our metric, we can tune GFP expression with a 20-fold dynamic range. And where we think this could go, one sector is the food industry, as there's a rising demand for plant and animal-based protein products. So we could apply our technology to boost the yields of these products, as well as alternative protein products such as those in the Impossible meats. Furthermore, because we're tuning the codons at an RNA or DNA level, we could also help improve the efficacy of messenger RNA vaccines that involve taking a piece of synthetic messenger RNA that encodes for the virus. We can codon optimize that to enhance the translation of the viral proteins that are meant to elicit an immune response, and hopefully make the vaccines more effective and potentially lower the doses needed. And as I mentioned, where we can not only boost protein production, but also decrease protein production, this may have applications in synthetic gene circuits, whereby you may want to turn off or suppress a gene to help boost the production of a subsequent downstream gene, and we could potentially apply our platform there as well. So ultimately, in summary, we've applied a variety of metrics, both systems-level codon analytics, tRNA metrics, and measures of translational regulation to engineer messenger RNA for protein production in your cells under your particular conditions. And we would love to chat at our poster. We are looking for an entrepreneurial executive to help us translate this venture into a spin-out company. We'd also love to chat about applications to food proteins and vaccines. And thank you for your attention.